Welcome members to TED Talks Education. We are still looking at biology, and in this episode we're going to focus much on regulation of enzyme controlled reactions. But before we continue much, what is regulation? This regulation would be the same as control. So enzyme controlled reactions are also controlled, meaning enzyme controlled reactions do not go up to infinity. They reach a certain stage and stop. When an enzyme is separated from salivary glands and salivary amylase comes to the mouth, does salivary amylase stay in the mouth for good? No. That salivary amylase is going to be what happens to it, is what we want to look at. The first one, when enzymes are done with the catalysis, they are degraded into amino acids and reabsorbed to make further proteins in secretory cells. So when salivary amylase is done with its work in the mouth, it will ascend, then it will descend to the stomach. Pepsin acts on it, breaks down into amino acids. We absorb it via the ileum walls into the blood system, which takes it to the cells that are capable of protein synthesis. That is one. Two, enzymes are only produced when there is a stimulus that stimulates the brain to instruct the secretory cells to start protein synthesis. So meaning secretion of enzymes is active because protein synthesis involves some energy during amino acid activation. Thirdly, and the last one, is called end product inhibition. End product inhibition. This one is a form of negative feedback which we shall expand later. Negative feedback. That in a series of reactions controlled by several enzymes, the final enzyme inhibits the production and action of the first enzyme in the cell. In other words, when a product, exact product is formed in an enzymatic controlled reaction, let's consider A being hydrolyzed, catalyzed to form B by enzyme, then B goes to C, and then C goes to D as our final product. So it will mean enzyme E1 that catalyzed this, and enzyme E2 that catalyzed this, enzyme E3 that catalyzed that. When this desired product is formed, the enzyme E1 is inhibited. So this is what we are calling the end product. So it inhibits the formation of enzyme E1 such that A can no longer be turned to B, and therefore production of E2 may not be possible. As I've said, enzymes are produced when there is what? A stimulus. A stimulus can be presence of food, presence of a substrate in a cell, so that the need now for production of that enzyme is there. We shall look at the control of digestion. It is all about smell, touch, thinking, and even the presence of food in the mouth that causes further circulation of enzymes in the body. The same, that when A is present, it will cause production of E1. E1 will catalyze the breakdown of the reaction of A to become B. Then the presence of B puts in place E1, enzyme 2. Enzyme 2 catalyzes the conversion of B to C. Presence of C leads to circulation of E3. E3 catalyzes the conversion of C to D. If D is our end product, that end product inhibits the formation of the first enzyme. When it does so, B won't be formed, E2 stops, E3 also will stop. So what is the significance of this? Significance of end product inhibition. The main significance is to ensure that chemical reactions in the body come to an end, so as not to exhaust metabolites and also save energy for other processes. Otherwise, if catalysis occurred throughout, a chemical reaction begins and it takes place throughout life, then the body would break down. So that is the significance. Briefly, let's talk about negative feedback in end product inhibition by illustration. Negative feedback mechanisms of control are those ones whereby a change that happens is what puts in place a corrective mechanism that puts it back to normal. So now, E1 is the one that has almost led to the formation of D, but presence of D now inhibits the formation of E 1. That's why it's called negative feedback. What is put in place is what puts away the parameter that put it in place. We shall expand it in our subsequent sessions of homeostasis to see how body systems are regulated. So when we meet again, we shall focus on another thing. Thank you.